So if you want. Good afternoon, everybody, or good morning, if you're in a morning time zone. It's very nice to see everyone beginning the year with the first Tika Talks. We're a bit late. We still have a lot of open spots for this year. So if you're interested in giving a talk here, please contact us through the Tika Talks email or hit us on Twitter. We will be glad to schedule a seminar with you. I apologize for the noise on my microphone. I'm on a random parking lot and there's cars passing by. So today we are having Dr. Marco Todesco, and it's great to have him here. He is doing incredible work understanding the patterns of coloration in sunflowers, and he will present a talk about his more recent paper. Before we start with the talk, just a few notes. We have our website, the Tika website at composite.org. In this website, you can see all the news about the association. You can register to be a member of Tika. And you can also catch up recordings from the previous Tika Talks. We have a YouTube channel where all the recordings are. We also have recordings of all the sessions from the last composite workshop at the botany meeting last year. So Jennifer just posted the link on the chat. And also at the end of the meeting, Jennifer is going to post a link to a conference called Plant Genomes Online. Did I get the name right? Okay. Which is a free conference that is going to happen next month in April. The abstract submission is already closed, but if you're interested in plant genomics, it's going to be a great assembly of speakers, and will probably be very worth it to attend. So before anything else, well, without further ado, I'm sorry, my brain is scrambled. Oh yeah, and Isaac just posted the link to the YouTube channel on the chat too. So please go there and check the past talks. We had an incredible assembly of speakers last year. So without further ado, Dr. Marco Todesco is a research associate at UBC in Canada, and he did his PhD in the Max Planck's Institute in Germany. I'm sorry, and he did also a postdoc at UBC. So you have the floor, and I'll stop talking because I can't say anything today.
Excellent. Okay, thank you very much for the introduction. I'll just share my screen. Okay. And okay, can you see it? Excellent. Okay, so as I was saying, thank you very much for the introduction, and thank you for the invitation to present today. I'm going to talk about some of the work we have been doing in the past few years on the genetic basis of adaptation in wild sunflower, and more specifically on patterns of ultraviolet pigmentation. Since this is a composite talk, I get to start directly speaking about our composite of choice, which are wild sunflowers. Wild sunflowers are, of course, a classic model system for studies of adaptation, speciation, domestication, for a number of different reasons, and I'm just going through some of them. So there's about 50 species of wild sunflowers that are native to North America, and many of them are interfertile, which makes for like a nice way to study what happens when you cross them. And the thing that is more interesting to me is that they grow really in a variety of different environments across all of North America. And some of these environments are also quite extreme. So they can grow on sand dunes, desert, salt marshes, serpentine soils, and so on. So there's a lot of raw material for studies of adaptation. The other nice thing about wild sunflower is that they are, of course, closely related to cultivated sunflower, which is a major crop. And the advantage of that, one of the advantages of that is that if we find something of interest or something potentially useful in wild sunflower, it is relatively easy to translate that directly into cultivated sunflower. And in fact, wild sunflowers have been used quite extensively in sunflower breeding in the past. And the other nice thing about wild sunflowers, and this is again partially because of cultivated sunflower as well, is that there's really a lot of genetic and genomic resources for this species, collections of thousands of accessions from all over the place, both for wild and cultivated sunflower. Many of these accessions have been resequenced, and I've just very briefly mentioned some of that in the talk. And then the other thing is that there is now a growing number of reference assemblies for cultivated sunflower and also for wild sunflower species. So this is the I think the latest count, which is of 16 reference assemblies. So these are all high quality, roughly not all of them, but most of them chromosome level assemblies. So there's currently 9 for cultivated sunflower and 7 for wild species of sunflowers. These are almost all annual sunflowers, although there is also an assembly for the varicatus, which is a perennial. And a few of those we generate them. So the Risberg lab, most of them have been generated by the International Consortium for Sunflower Genomics, of which the lab is part, but is also which is led by Stefan Munoz at INRAE. And we're also working on adding a couple of Elian Tosano's reference assembly, as well as Biguillera dentata as an outgroup to help with the analysis of these genomes. So this is all they're all relatively new. They're not really, I think, publicly available yet, but they will be soon. And as soon as they are, I think it's going to be a fantastic resource for studies in sunflowers and in general comparative genomics. But as I said, so my main interest is in adaptation and the genetic basis of adaptation. And one trait that is almost quintessentially adaptive is flower color. Flower color, patterns of coloration, and other characteristics of the flowers are generally due to the coevolution of plants with their pollinators. So pollinators are really what is driving most of this diversity. And this results in the fantastic variety that you can see everywhere. Most of the diversity, of course, is between different species, but there is often also diversity within one species. So plants from the same species will have very different colors or patterns. However, this seems to be one trait where sunflower doesn't really have much to contribute, because all wild sunflowers are uniformly yellow. So the legals are just yellow and yellow throughout the whole legal. And that is indeed the case for the human eye. So to us, these two sunflowers look basically exactly the same in terms of color. But many insects and many animals can see light in the ultraviolet range. So to them, these sunflowers will look quite different. In fact, what sunflowers accumulate UV absorbing pigments at the base of their legals, and across the whole inflorescence, this usually creates this kind of circular pattern, which is called a UV bullseye. Patterns like these or other patterns of ultraviolet pigmentation are quite common in flowers, and they seem to be ubiquitous in wild sunflowers. So these are, I think, 15 or 16 different species and subspecies of wild sunflower. And you can see that in all of them, you have at least some level of UV pigmentation. And the reason why UV patterns are so widespread is because pollinators like them. So people over the years have done a lot of different experiments manipulating these UV patterns, things like using sunscreen or cutting and pasting petals to make different patterns, as in this example here. And what they found is that basically having these UV patterns make the flowers much more attractive to pollinators. So you have much more pollinator visits, which of course it's good for the flowers. Just so for the remainder of the talk, this is like the standard way in which UV patterns are measured in flowers with circular symmetry. So like sunflower, they are measured as UV proportion, which is basically just the length of the legal or over the petal that is UV absorbing versus the complete length of the legal. So if you have high value close to 1 of this legal UV proportion, means that most of the legal is going to be UV absorbing. So this pixel will look mostly black. Low level, instead, low value close to 0 means that there's really just a very little amount of UV bullseye at the base of the legal. And most of the legal will be UV reflective, so kind of paler color in this picture. The other thing you probably have noticed from this picture is not only that UV patterns are everywhere, apparently in sunflowers, but also that there is a lot of diversity in the size of these patterns, right? You have everything from very little UV absorbance to apparently everything being UV absorbing. And that's something very interesting to us, so we want to understand why that is the case, and how this diversity is controlled. And we took advantage of some resources that we were generating at the time. So this is part of a larger project aimed at understanding diversity in terms of both phenotype and genetics in wild sunflowers. So in the summer of 2015 and 2016, Dylan, a former postdoc in the lab, took his red pickup truck and drove across most of North America, so from southern Canada to northern Mexico, and visited hundreds of population of wild sunflower, collecting seeds from thousands of species, and send them all back to Vancouver for us to work on. He focused on three plants, three species. The first one is Elianthosanos, so the common sunflower, which has the broadest distribution and is, of course, the closest relative to cultivated sunflower. The other species is Elianthus petiolaris, which also has a fairly broad distribution, prefers sandier soils. It has adapted repeatedly to growing on sand dunes. And then the third one is Elianthus argophyllus that grows only in the southern Texas. So in the southern part of Texas, it's called the silverleaf sunflower, and is sister species to Elianthosanos. So as I said, Dylan sent all the seeds. We grew them all in a common garden experiment, and we phenotyped them fairly extensively, well, very extensively. So we took developmental and morphological measurements for all of them. Things like carbon, nitrogen content in leaves. We have a collection of about 80 phenotypic traits for 1,500 of these plants. So from 150 of this population, we took 10 plants each and grew them in a common garden. And we have, of course, also climate variables for the population of origin of these plants, and also Dylan collected soil. So we have chemical data about the soil these plants initially grew in. And what we found is that there is really a lot of diversity, not only between species, as I've shown you before, but also within species. And in particular, in Elianthosanos, you have everything from inflorescences that are almost completely UV reflecting to inflorescences that are almost completely UV absorbing. And when we saw this, we had, of course, a couple of questions that immediately came to mind. The first was, how do they do it? So what are the pigments that are responsible for these UV patterns? What are the genes and the genetic pathways that are underlying this diversity? And the second question is, why would you do it? So why do you have this diversity? What are the drivers, if any, of this variation? Answering the first question was relatively straightforward. So we had not only a phenotype of these 1,500 plants that we grew together in Vancouver, but we also resequenced them. So we have extensive genotypic data for all these plants. So there's a lot of diversity between and within. Yes. Wild sunflower species. So we had hundreds of millions of genetic variants that we could sequence. And for all of these three species that I mentioned before, we in the end extract these sets of more than 4 million very high quality SNPs. This is a phylogenetic tree that is based on part of this data. It doesn't really tell much, it's just to show that everything is as it's meant to be in this data set. So here you have Elianthosanos with cultivated sunflower nested in. This is Elianthus argophyllus, which is the sister species I was saying before. And down here you have Elianthus petiolaris, which is divided into fairly separate subspecies. So we have genotype data, phenotype data, and we put them all together in a GWAS, so genome-wide association mapping experiment. And since the two petiolaris subspecies are so different, we did the analysis separately, which has the problem of reducing a bit the power of our analysis because the populations are smaller. We didn't find any significant association for one of the subspecies, Elianthus petiolaris phallax, but we did find some associations that are above the significant threshold for Elianthus petiolaris, and so there's a few different loci, which is what you would expect for a quantitative trait. When we look in Elianthosanos, though, what we found is that there is one locus on chromosome 15 that explains really about 2/3 of all the diversity that we see for patterns of UV pigmentation in sunflowers. Which is always very nice when you have one of these rare GWAS that work quite as well. When we look closer, we found that the SNPs that have the strongest association with floral UV patterns are about 5 kb upstream of MIB111, so in the promoter region of this gene. And this SNP, so this SNP that I might probably mention later on in the talk as the chromosome 15 SNP or the chromosome 15 UV SNP. So this SNP has really quite a strong effect on the patterns of UV pigmentation in legals. So if you have one of the alleles of this at this SNP, so what we might call the small allele, so plants that are homozygous for the small allele have about 1/3 of the legal that is UV absorbing. Plants that are homozygous for the large allele at this SNP have about 3/4 of the legal that is UV absorbing. And this seems to be a completely additive trait because plants that are heterozygous are have on average an intermediate phenotype. So what does HA MIB111 do? So the arabidopsis homologue of this gene, MIB111, is part of a small family of transcription factor. So it's 3 transcription factors that are also called production of flavonoglycosides or PFGs. And as the name kind of implies, they are important for production of flavonoglycosides, which are a type of flavonoid. So what this gene do, what this gene does is to control expression of a number of enzymes that are in the biosynthetic pathways of flavonols. Flavonoglycosides, as I said, a kind of flavonoid, and they have been involved with a number of responses to different stresses, both biotic and abiotic. But perhaps more relevantly at this point, they are the UV absorbing pigment in several plant species. So they make for a pretty good candidate as a UV absorbing pigment in sunflower as well. And indeed, when we checked legal cell plants with large and small UV patterns, we found that most of the UV absorbing pigments in legal cell plants with large UV patterns are flavonoglycosides, so these gray peaks here. And when instead look at plants with small UV patterns, we found basically that there is no accumulation of any of these compounds, suggesting that indeed these are the UV absorbing pigments of sunflower inflorescences. And that's true not only when you compare plants with different patterns, but also within a same legal. If you compare the base, which is UV absorbing, and the tip, which is UV reflecting, you can find a difference in the accumulation of these flavonoglycosides. So in gray, you have the so these are all UV chromatograms. So we extracted pigments from the legals, and you can see that the UV absorbing pigments here, the flavonoglycosides, are present at the base of the legal, but they're almost completely absent at the tip of the legal, suggesting that MIB111 probably regulates patterns of UV pigmentation through its role in accumulation of these UV pigments, flavonoglycosides. At this point, the ideal way in which you would confirm this would be to take different alleles of MIB111 in sunflower and put them in different sunflower lines and see if you can recapitulate plants with large or small UV patterns. Unfortunately, sunflower is not very friendly to transformation, so we have been spending a lot of time, and a lot of other people have been spending time trying to optimize the transformation protocol for sunflower, but we're still working on it. In the meanwhile, we did the next best thing, which is heterologous complementation in arabidopsis. When we look at where MIB111, so the arabidopsis version of this gene, is expressed, we found that it is expressed at very high level, especially in petals. And when we look at petals, we found that indeed petals of arabidopsis absorb quite strongly in the UV, which was quite of a surprise because, as I said, these UV patterns normally are associated to pollinator attraction, and arabidopsis is really largely a sulfur. There's very little in the wild. There's very little pollination, so very little intercrossing has been detected. So yeah, I wouldn't have expected them to have any kind of patterns, but there they are. And yeah, the nice thing is that in the mutant, in the arabidopsis line that is a mutant, so a loss of function mutant for MIB111, you lose completely these UV patterns. And this correlates with patterns of accumulation of flavonoglycosides. So in green, you have the wild type, Columbia, and you have that the UV, this is the UV range. You have all these green peaks that are flavonoglycosides, and in the mutants, these peaks are accumulated at much lower levels. And when we take the sunflower gene and put it into the arabidopsis mutant line, we can recapitulate the phenotype of the wild type and also the accumulation of these flavonoglycosides. This is the case both when we express this gene, the sunflower gene, under a constitutive promoter, so it's expressed everywhere at very high level, but also when we use the endogenous promoter of the arabidopsis version to drive the sunflower version of MIB111. we have basically the same result. So you have you recover this UV accumulation of UV pigment, and you have also accumulation of flavonoid glycosides. The other interesting thing that you can see from this figure is that we use two different versions of MIB111 from different sunflower lines. One version that has the small allele, so the small allele, that particular SNP I mentioned before, but also plants that have small UV patterns, and then the version of MIB111 from plants with large UV patterns, and both of them seem to really have the same effect when they are expressed in arabidopsis, suggesting that the difference between alleles of MIB111 that result in small or large UV patterns is not really due to difference in the protein itself, so in the gene itself, but it's probably something at the regulatory level, which is also consistent with the fact that the strongest association that we found in GWAS is in the promoter region of MIB111 and not in the gene itself. So where is MIB111 expressed in sunflower? Similarly to arabidopsis, it's really mostly expressed in legals. So this is RNA-seq data that were previously published. But then when we look more closely at the sunflower legals, we found that it is expressed specifically at the base of the legal, where the UV pigment is accumulated, and not at the tip. And this level of expression are particularly high in developing legals. So when the legal is turning from green to yellow to the human eye, that's also when the expression levels of MIB111 is highest. And that makes sense because, of course, you would want this expression to be high early on when these UV patterns are being established. So we found that MIB111 is expressed at high level at the base of developing legals, and that's the case also for FLS1, that stands for flavonoid synthase 1, which is a gene that is involved, of course, in the synthesis of flavonoid glycosides, and it's also directly controlled by MIB111 in arabidopsis. And then finally, the last thing we checked was expression levels across a larger panel of wild sunflower lines. So we had 46 lines with either large or small UV patterns. Check the expression level for MIB111 in developing legals, and we found, as we would probably expect from these experiments, that plants that have larger UV patterns have also higher level of expression of MIB111. Again, suggesting that the difference between alleles is at the level of the promoter of how the gene is regulated, and not really about the gene function itself. And then we spend a remarkable amount of time trying to figure out exactly which sequence differences are responsible for this difference between alleles. And the short story is that it's complicated and we don't really know. But we did learn something. So here you have three versions of the whole locus. So in yellow is the gene itself, and here there is the regional promoter that is closest to the gene. This is the SNP with the strongest association in the GWAS, and here there is a region of the promoter that is further away from the gene. Oh, sorry. And so what we found is that cultivated sunflower, the reference sunflower has an allele that is fairly similar to wild sunflower that has large UV patterns, and indeed cultivated sunflower, as you'll see in the next slide, have in general fairly large UV patterns. But while there is a lot of variation between different lines, so we sequence about 15 or 16 different wild sunflower lines at this locus. While there is a lot of diversity between these lines, there was really no pattern that stood out in terms of plants that have large UV guides having one SNP or one structural variant, and plants that have small UV patterns having a different one in the gene itself or in the proximal region of the promoter. However, the situation is very different the further you go away from the gene. So in the region upstream of this let's say causal SNP, of the SNP with the strongest association, there's really a lot of structural and sequence variation that differentiates plants that have large and small UV patterns. That's nice, but of course, since there's a lot of variation, we cannot really pinpoint which one is the variation. I'm hoping that some of the reference assemblies, or the new reference assemblies I mentioned before, might help a bit with this, but for now, we just know that there's sequence diversity, but we don't really know how this affects the expression of MIB111. And as part of this exercise, we also sequence a couple of so the MIB111 locus from a couple of other wild sunflower species, so Argophilus and Petiolaris. And it turns out that these species have instead version of the gene that are very close to wild annus plants that have small UV patterns. And indeed, when we look at, so we had resequencing data for these 1,500 sunflower species that, as I mentioned before, when we look at the genotype at this chromosome 15 SNP across these plants, we found that the correlation that we see between genotype at this SNP and UV patterns in wild annus is really preserved also across different species. So Petiolaris, Argophilus, and Niveus, so 3 species of wild sunflowers, they are either fixed or almost fixed for the small allele at the SNP, and they all have fairly small UV patterns in their inflorescences. And on the contrary, instead, cultivated sunflower is almost fixed for the large allele of the SNP, and it has fairly large UV patterns. Taken all together, this hopefully shows that the difference, so the answer to the first question, how is this variation achieved in UV patterns? And the answer is through cis-regulatory variation in hamiband 11 that affects how flavonoid glycosides are accumulated in the legals. And then we went to the next question, which is why. So why would you have these different patterns? As I said, the main driver for differences in any color pattern, but also in particular UV patterns, is thought to be pollinators. So that's why this pattern exists in the first place. And we wanted to see also if, like, not only pollinators like to the presence of these patterns, but also they can differentiate between different type of UV patterns and if they have preferences. So we started making a very straightforward experiment. We took 2 populations that had roughly the same flowering time and flowers of more or less the same size. Put them out in the field, put flowers one next to the other, and counted how many bees and bumblebees visited either flower. And we found that there is a fairly significant difference between different inflorescences, with inflorescences with larger UV patterns being favored by pollinators, which is nice. But of course, these are just two species, two plants or 2 populations. There could be many other differences between their flowers that we cannot see, say content of nectar or something else that might be responsible for the difference that we see. So we try to account for some of these other factors by repeating this experiment in a much larger field. So we have about 1,500 wild annus growing in a field here at UBC. We selected 84 plants that flowered at the same time and had roughly the same number of flowers, and we checked their UV pattern, the size of their UV bull size, but also how many pollinators visited them over a period of a few days. And we found that pollinators seem to prefer intermediate to large UV patterns, and they really dislike plants that have very, very small UV patterns, and that these very small UV patterns are also quite rare in the wild, which might be why they are. So if the pollinators don't like them, there's probably some selection against them. And this is consistent to what other people has found, again, with some of the artificial manipulation that I mentioned before. For example, here in Rudbeckia, you can see that so they cut and pasted different petals to make different patterns, and in this case as well, larger UV patterns attract more pollinators, while smaller UV patterns attract less pollinators. So this is nice and shows that there's a difference, there's a preference of pollinators for one or the other of these patterns. But if it's true that pollinators prefer larger patterns, why you have different patterns, right? You would expect that everything would be large. And this is also further complicated by the fact that this diversity in UV patterns is not randomly distributed across the range of wild annus, but there are some fairly well-defined geographic patterns. So smaller UV bull size are common in southeast of North America, while very large UV bull size are common in either central US or California. This difference could be due to differences in pollinator assemblages or pollinator preferences in different areas. However, this seems unlikely because flowers really doesn't have one or few specialized pollinators, but it's very broadly pollinated. Right? So it seems unlikely that you would have such well-defined differences across many different species of pollinators across these large areas. And the other thing is that many of the major pollinators of sunflowers are ubiquitous across North America. So they will be found in both in areas with large and with small UV patterns. This doesn't mean that differences in pollinator assemblages do not play a role, but it was enough to kind of interest us in looking for other possible explanations. And this is partially so what we look at was whether some abiotic factor might drive this difference. And this is for two reasons. One, because of this kind of well-defined geographic patterns, which might be tracking some kind of climate variable, but also because, as I mentioned before, flavonoglycosides are known to be involved in responses to different kind of stresses. We focus on three factors in particular for different reasons: UV radiation, temperature, and relative humidity. So we started with UV radiation because it kind of seemed to make the most sense intuitively. UV radiation is, of course, damaging for plant cells, and flavonoglycosides absorb UV radiation, so they basically work as sunscreen, and they protect the cells. And it has been shown in Argentina and Serena that that's indeed the case. So in this plant, you have larger UV patterns in the flower the closer it gets to the equator, so the higher is the level of UV radiation. And that's thought to be because these UV patterns absorb UV radiation that otherwise would be reflected to the anthers and damage pollen. And indeed, they could show, like, so this is a paper from Koski and Ashman from a few years ago. They could show that in the presence of UV radiation, larger UV patterns result in higher pollen viability. So meaning that really, it seems like that's one of the main roles of UV patterns in this species. However, this seems unlikely to be the case in sunflower because so Argentina and Serena has this bowl-shaped flowers, so it makes sense that radiation will be reflecting into the anthers. Sunflowers have a much flatter inflorescence, so it seems unlikely that any significant amount of UV radiation will be reflected from the ligos to the disc flowers. And indeed, when we look at associations, correlations between size of UV patterns and the intensity of UV radiation across the range of wild animals, we found basically no correlation. So it's not UV radiation, most likely. The next thing we checked was temperature, and that's for two reasons. The first is that there is a very good correlation between size of UV patterns and average summer temperature, with larger UV patterns being more common in colder areas. And the other reason is that it is well known that inflorescence temperature is very important for pollination in sunflower. There have been a couple of studies in the past few years showing that the reason basically why cultivated sunflower faces east in the morning once it's fully open is because it will warm up faster, and warmer sunflowers are more attractive to pollinators. So you see here, these are cultivated sunflower facing east, they get a ton of pollinator visits. Cultivated sunflowers facing west, so away from the sun, in the morning they get basically no visits. And then when they artificially warm plants, inflorescence is facing west, you get quite a strong increase in pollinators. So it's really, temperature makes a lot of difference. So I set up everything. I went up in the field at 5 in the morning to take thermal images across the whole morning as flowers, inflorescences with large and small UV patterns got warm up. And unfortunately, we saw no difference whatsoever. So these are some of the results. So we did a few different time series, and you see on the left you have always the plant with the small UV patterns, large UV patterns on the right, and there really no visible difference in the temperature of the ligos, regardless of the UV patterns. And that, in hindsight, that makes sense because UV radiation is not particularly energetic, so it only represents between 3 and 7% of the total energy that comes from the sun at Earth level. So it probably shouldn't warm up these inflorescences much. One thing that you can notice though is that the disc does seem to get a bit warmer in plants that have small UV patterns. However, that has nothing to do with the ligos. So we did a stupid experiment in which we took away all the ligos and checked the temperature, and regardless of whether they have or not ligos, you still see that this particular line gets, the disc gets warmer. So it's something else that is not ligo related. Interestingly, when you remove the ligo, you remove also the difference in pollinator preferences between these two lines, but that's probably just because there's no pollinator whatsoever that goes to any of these plants. So not surprisingly, the petals or the legals are really important for attracting pollinators. We did also another version of this experiment where we just checked individual legals. We put them in the sun for 15 minutes and measured their temperature. These are different pairs of legals that we put out from different populations. And as you see, there's really no difference between plants that have large or small UV patterns. So temperature is not really, that doesn't seem to be, at least, so the effect of temperature on the legals doesn't seem to be relevant in this case. And then finally, we look at relative humidity. And what we found is that this is the climate variable that has the strongest correlation with UV patterns. So you have larger UV patterns in areas that are drier. And this is relevant because at lower humidity, you have more transpiration, so you have more loss of water from the flowers, from plants in general, and from the flowers. And it is known that flowers are a major source of transpiration and water loss. So much so that at least in some species, you have so plants in different populations have been selected to have smaller flowers in drier area. The other relevant piece of information is that it has been shown in arabidopsis that overaccumulation of flavonoid through the overexpression of MIB12, which is one other of these PFG, production of flavonoid transcription factor that I mentioned at the very beginning. So plants that overexpress this gene have a lot of flavonoid. They are much more resistant to drought. As you can see here, there is wild type after 14 days without water, and these are plants that have more flavonoid. And this is due to a reduction in water loss. So they're able to retain water better and limit transpiration. So we thought we would check that as well. And we took detached legals from plants with large and small UV patterns. We tried to cover as many different populations as possible to kind of normalize other possible differences between these lines. And what we found is that legals that have larger UV patterns, so they accumulate more flavonoid, they lose water at a significantly slower rate than legals that have small UV patterns. And this is not like a general difference between these plants. So one possibility would be that plants that have large UV patterns come from drier area, so they are overall more they lose less water and more able to retain water. When we did the same experiments, instead of using legals, we used leaves from the same plants. We found no difference whatsoever in the rate at which they were losing water. So this is something that is specific to legals and not a general trait of these particular plants. So it looks like really it could be that this difference in relative humidity and so the ability of the plant of being able to lose less water and retain more water are really one of the factors that might that can drive these differences in UV patterns. However, of course, transpiration is not overall bad. It's very important, obviously, to drive water movement within the plant, but it's also important for protecting plants from overheating. So when you have transpiration, you have evaporative cooling, so it prevents organs from becoming too hot and to be damaged. And so it will make sense that this might explain why you have these small UV patterns in areas that are hot and humid, where preserving water might not be quite as important, but protecting against high temperatures might be a driving factor. And the fact that both temperature and humidity might be important in driving diversity in UV patterns is also consistent with the fact that when we did a GA, so this is a genotype environment association analysis, it's very similar to GWAS, just that instead of looking for associations between phenotypes of individual plants and the genotype of individual plants, you look for correlations between association between environmental variables or other characteristic of the whole population and allelic frequency in that population. But the idea is the same. So we look basically for regions of the genomes that are associated with different environmental variables, and we found that there is a significant association for both average temperature and relative humidity in the promoter region of mid-boundary level, suggesting that both these variables are probably important in driving this diversity. And the way this interaction goes is, as I mentioned before, so hopefully this plot can be understood. Basically, when you have low humidity, so this dashed line here, temperature doesn't really make as much of a difference in terms of UV patterns. So you always tend to have rather large UV patterns. But then when humidity is higher, so when water might be less limiting, temperature really makes a lot of difference. So in colder places, you have large UV patterns, and in warmer places, you have small UV patterns. So this answer to a certain extent the second question. The answer is, of course, so to the question, why is there such variation? The answer is it's complicated. So it's a combination of abiotic and biotic factors. So abiotic factor being humidity and temperature, and biotic factor being pollinator preferences. That really seems to drive this diversity and the patterns of diversity we see across North America. And then just to conclude, one question that is quite natural, I think, is this useful in any way? Intuitively, yes. So of course, both pollinator and the ability of retain more water are important from an agronomical perspective. So they could be useful traits for cultivated sunflower. And what we have what we have found, that you have seen already before, is that indeed cultivated sunflower already has it's already almost completely fixed for the large version of the hamiband 11 gene, which confers larger UV patterns. So increase pollinator attraction and also decrease transpiration, decrease water loss. This is probably not something that has been selected directly. I don't think any breeder has ever looked at UV patterns, but it can be that unwittingly, these traits are favorable, so this allele has been selected. That said, there is still, of course, a lot of variation in UV patterns within cultivated sunflower, so there's still a lot of room for improvement and optimization depending on where you want to grow your cultivated sunflowers. With this, I conclude. This is, of course, like a large collaborative project. Here on the left, you have all the people that have been involved more directly in the UV pattern project, which was basically built on the back of a larger effort that I mentioned in the beginning to understand adaptation, phenotypic variation, and genetic variation in wild sunflowers. With this, I conclude, and I thank you all for your attention. I'm very happy if there is any question.
Thank you so much, Marco. That was very good and very interesting. If anybody has questions, you can either speak your questions in the microphone or put them in the chat. And I'll ask Isaac to read the questions on the chat for me, because I'm on my phone, so I have trouble checking the chat and staying in the call at the same time.
No problem.
Thank you.
Thank you, Marco, for an excellent talk. There's a question in the chat from Dr. Schilling. He asks, does the type of flavonoid make any difference? Different wild sunflowers have chalcones versus flavanols.
It's hard to tell. So we only find flavanols. There's no chalcones or arones in the wild sunflower, at least in Annos and Petals, where we are looked at. It might be. I don't think it would make much of a difference in terms of pollinator attraction, because that's just it's a visual cue. It might make a difference in terms of effects on transpiration. That might be something specific to flavanols versus other pigments. It is really not well known how flavanols are affecting transpiration or water loss. So that's something that we'd like to look a bit more into it. But right now we don't really have a straight answer.
Great. And there's another question here from Jen Mandel. Jen, if you want to ask your question yourself. No? Okay, then I'll read it. How about tests for selection on these genes?
Yeah, we haven't done any for a very technical reason. So the problem is that this gene is in a very repetitive area of the genome. So the promoter region basically there's big chunks where we don't have any resequencing data, and that's the problem with short read data. We couldn't map anything uniquely there, so there's big areas where we don't know, and that's why I think the strongest association we found is that in this SNP. I don't think it's because necessarily that SNP is the closest to the causal locus. It's just basically the closest SNP we can genotype to that messy area where we have structural variation and sequence variation that can't really be genotyped. And the problem is, of course, both the repetitive nature of the region and also the fact that we rely on reference genomes that are cultivated sunflowers. So those regions are very different. There might be things in there that are not there in cultivated sunflowers. So that's why I'm hoping. So we are right now, so we already sequenced them. We're trying to assemble 2 wild anodes that are hopefully fixed for the large and small allele of this. That's not the only reason why we sequence them. That would be an expensive kind of thought experiment. But the idea would be to be able to assemble the whole region and really see what's in there. I mean, at this point, we think it's structural variation in promoter. It could be, who knows what's in there. It's been very hard. So I spent 1.5 years basically trying to amplify and piece together this locus. Very disappointing, but kind of work.
Jen followed up with a question, which was, what about synteny with other genomes like lettuce or? Yeah,
that's a very good question. We haven't checked, and I don't really know actually. Yeah, I don't know if it's known how much variation there is for UV patterns in lettuce, for example. But that would be great. Yeah. So I think that would be part of, once we have all these genomes, kind of do a more comprehensive analysis.
So a question from Camille. Very interesting talk. I'm curious about the technical side. How do you document UV patterns in the field? Do you need special equipment?
The only special equipment you need is a UV camera, or a camera that has been modified to basically be able to see UV radiation. What we have is a regular Canon D40 that has basically, usually there is a filter, so UV radiation doesn't go through the sensor. The filter is removed, and then you put an additional filter that keeps out everything that is not UV. And the rest of the setup is a tripod and some white paper sheet where you stick the deagles. We haven't had like much luck using artificial UV sources for doing these pictures. So the best way is to do it really in natural light, which is problematic in Vancouver because, yeah, natural light limited at times.
I had a question as well. I was really interested in the last part of your talk where you're talking about why there's this variation to begin with. And you showed kind of the association where it looks like, mainly those populations in South Texas or in the more arid part of the range express less UV. And I was just wondering, when you look at it in terms of the phylogeny of the wild sunflowers, whether is that basically like one clade? Is that one group that is expressing that, and does that tend to be an ancestral or a derived trait within the phylogeny? Yeah,
so in the phylogeny, there's one slide that maybe goes. Sorry, I'll have to scroll through all of them. Very annoying. But the short answer is it doesn't seem to be really very clear separation in which plants have large or small UV patterns. The problem there also is, of course, and so a lot of the wild species were actually, sorry, almost there. Yes, so here, this is my attempt to kind of represent UV patterns on top of a phylogeny. So the size of the black dot is basically the size of the UV absorbing part. And as you can see, it's a bit all over the place. The one limitation is, of course, that we only have images for 1 or 2 plants for most of these species. So it could also be that there is a lot of intraspecific variation in these other species as well. So it's kind of hard to infer. We would like to be able to complete this work and also extend it to more individuals for each species and see if it's the same pigments as was mentioned before, if it's the same gene that is involved.
Really cool. At this point, if anybody wants to unmute and ask a question, there's no other questions in the chat that I can see.
I think
there's one more.
Yeah.
Oh yeah. Morgan.
Yeah, I can ask. Great talk, Marco.
Yeah, thank you.
I actually asked the same question of a colleague last week. She's setting up across a bunch of private property in Texas, cameras for long-term imaging for a phenological study. And I was curious if she had any UV filters for cameras, and she said she didn't. She couldn't find any that would meet her needs. So she's setting up maybe 80 cameras, And the idea is to take images at the exact same time each day for a year. And so I guess to piggyback on Camille's question, do you know of any options that might be able to automate the dual sort of typical camera?
Yeah, I fear it's a very, I mean, it's a very kind of niche field of photography. So it's either cameras that have been manually altered by selecting few people that know how to do that, or very specialized equipment that is extremely expensive. I don't think there is something similar to a field camera that would do that. Not that I know of, at least. Unfortunately, it's it's hard to automate that level.
Cool. Okay, now that makes sense. Maybe if she has she does have a little extra money, maybe she could set up a smaller handful of sites where she would install those cameras. Cool. Thanks.
Cool. Well, unless there's any other questions, I think we can give a virtual applause to Marco.
Thank you.
Thank you for that really amazing talk. And looking forward to future TICA talks. This talk has been recorded, so you can share it, and it'll be on the YouTube very soon, and you can send it around to other people who you think may find it interesting. So thank you everybody for joining, and we'll see you at the next TICA talk.
Thank you.
And before we leave, the next TICA talk is going to be on May 9th. So the second week of May. And thank you so much for coming. Thank you, Marco, for accepting our invitation. And see you all again on May. And visit our website and YouTube channel.
Bye.